All right, welcome to another video on heart anatomy. Today, the primary topic is action potentials, and the heart throws a couple of curves at us. It's got a couple of different cell types, and they have different action potentials compared to what we're used to. So maybe the first thing we need to do is just quickly review what a typical action potential looks like, and by typical I mean a standard neuron or even skeletal muscle when we're talking about the neuromuscular junction. What does that action potential look like? So if we were to graph it out, there's a couple of things that we can remember, just as quick review. We start at resting membrane potential. Of course, resting membrane potential is established by, first and foremost, the sodium-potassium pump that creates gradients, and this is followed by increased potassium permeability, primarily through leak channels. As potassium leaves, the cell gets more negative. And so for a typical neuron, we consider resting membrane potential right around minus 70 millivolts. And we're measuring this charge inside the cell. So we're sitting here at resting membrane potential. Then something happens, and we start to depolarize. This depolarization event, for a standard neuron, this could come through maybe ligand-gated channels. As an example, and a very common example, a neurotransmitter binds to a ligand-gated sodium channel and opens it up. Sodium goes in and starts to depolarize, right? This is how neurons talk to each other. It could also be mechanical. These channels we see in sensory, if it's talking about touch or even hearing, this is all part of how some sort of pressure exerts a force on these mechanical channels, opens them up, cations rush in, and the cell starts to depolarize. The trick, of course, is hitting a value that we call threshold. What's so special about threshold? If you remember, this is when the voltage-gated channels open. And for this action potential, we would see a massive spike. We're going up. This is all driven by voltage-gated sodium channels. These are rapid channels. They're kind of on a timer. They open and then they close. And of course, when they close, we stop going up. Now, what happens? Well, we want to go back down. So this repolarization event, and oftentimes would hyperpolarize and then come back to resting membrane potential. This, if you recall, is all driven by potassium. So this is when the voltage-gated potassium channels become fully open. As potassium leaves the cell gets more negative. Down here at the bottom, right, where we've hyperpolarized, the permeability is still driven not only by the leak channels, because they're always open, but also still these voltage-gated potassium channels. And we see that as we increase potassium permeability, we get very negative. As these voltage-gated channels close, then the permeability is limited once again to the leak channels, and we come back up to resting membrane potential. That's a typical action potential that we've learned about before, and it's fairly easy to understand. Now what we want to do is add our knowledge and see if we can figure out what's going on in the heart. Of course, in the heart, there are two different types of cells. Let's talk first about pacemaker cells. Pacemaker cells, sometimes people think, well, pacemaker cells, these autorhythmic cells, they're only hanging out in the pacemaker. That's not actually true. If we consider the heart for a moment, there's the heart. Of course, it's two different pumps on the left side and on the right side. If we follow the electrical conduction pathways, and we'll talk more about these later, we start right here in the SA node. This is what we consider the pacemaker. But we also have rapid conduction pathways traveling down to the AV node. And then from the AV node, we have the bundle of His that then divides up into bundle branches, go down the septum, and these in turn swing up, and this is where we have the Purkinje fibers. We also have a signal. This is Bachmann's bundle coming over. It's a rapid conduction pathway to deliver the signal, the action potential, to the left side of the heart. Everything that we see up here, the SA node, the AV node, the Purkinje fibers. These are all made of pacemaker cells, and they actually all have their inherent pacemaker potential. Of course, there's differences. The SA node tends to tick at about 80 beats per minute. The AV node is about 40, and then the Purkinje fibers are about 20. But all of these structures, SA node, AV node, Purkinje fibers, they all have inherent pacemaker activity because they're all made of these pacemaker cells. Okay, so with that in mind, what does this action potential look like? If we graph it out, there's a couple of things that I want to point out. Number one, there is no resting membrane potential. Well, that's super interesting. I'm not saying that I don't have potassium permeability and all of that stuff is still there. But the reason these are special is because they don't require any external cue to fire. Remember, when we were talking about the neurons above, they required an external cue, whether it was a ligand-gated channel opening or a mechanical channel, something to get them to go. But that's not what we see here. We see rather, if we were to draw this, we see these dipping down, but just turning right back up by themselves and heading back up. There's still a threshold. We can draw that in here. There's our action potential. The first thing that I want to look at is this region right here. This is the pacemaker potential. And interestingly, it's driven by a very special channel that we call a funny channel. The funny channel is actually a nickname for the HCN channel. This stands for hyperpolarized activated, and the CN stands for cyclic nucleotide gated. So this is a hyperpolarized activated, cyclic nucleotide gated channel. Funny refers to this. What does that mean? Right, when we looked at the neuron, the voltage-gated channel opened above threshold. These are funny simply because they're backwards. These open up below threshold. So if we come and explore that in our picture, we see right here, as soon as I get below threshold, what happens? These funny channels open and sodium starts coming into the cell. And as sodium comes in, what happens? I just turn around and start to depolarize once again. Now, what about the cyclic nucleotide gated? I'm going to come back to that in just a second, but let's for now walk through the rest of the action potential. The action potential itself, once we hit threshold, if we're contrasting it to the previous action potential from the neuron, this one's different because this one's actually driven by voltage-gated calcium. These are T-type, T stands for transient, voltage, T-type, voltage-gated calcium channels. Now that's interesting. Mechanically speaking, these types of channels work a lot slower than the rapid voltage-gated sodium channels that we see before. So it's a little bit above and beyond, but this does play a little bit into understanding, number one, the important role that calcium plays in the heart, and number two, how timing of the pacemaker potential is very, very important. Of course, the backside of this is very similar to everything else we see. So we've got voltage-gated potassium. This is all potassium over on this side, just like we see before. So now that we kind of have the understanding of the ions that work here, I want to come back to this cyclic nucleotide gated business. The cyclic nucleotide that we're interested in especially is cyclic AMP. And you might recall from G protein coupled receptor signaling, in a fight or flight response, what happens to the heart? The heart rate goes up. The hormone driving the fight or flight response is epinephrine. So let's see if we can't figure out how this works. Epinephrine from the adrenal gland, or if it's just local sympathetic activity, it would be norepinephrine delivered directly to the heart. This is going to bind to beta adrenergic receptors on these pacemaker cells, these are G protein coupled receptors. And if you remember from G protein coupled receptor signaling, the ligand binds, we activate the alpha subunit. The alpha subunit, in this case, goes and activates adenylate cyclase. And with adenylate cyclase activation, the job of that enzyme is to take ATP and convert it to cyclic AMP. Now we've made our cyclic nucleotide. That's what epinephrine does to these cells. So once we have cyclic AMP, well, remember, cyclic AMP regulates the gates on these funny channels, cyclic nucleotide gated. Cyclic AMP binds directly to the channel and increases sodium permeability. So when cyclic AMP binds, we have increased sodium coming into the cell. Think about for a second what that's going to do. If we have increased sodium, I'm going to try and overlay this on the graph, then we're going to turn around a lot faster, and we're going to come up a lot faster because there's more sodium coming in. Of course, the rest of the action potential is the same. This is driven by voltage-gated calcium. None of that changes. So, the rest of this happens the same. But I want you to recognize, right, the difference in the slope between this line and this line. As I increase the slope of the pacemaker potential, the heart rate goes up. Look at the difference between these peaks. This represents a heartbeat. The gap from here to here essentially represents my heart rate. But with the addition of epinephrine to cause the production of cyclic AMP, which then binds to this funny channel and increases sodium permeability, by increasing the slope of that pacemaker potential, look what we've done. We've decreased the distance between those peaks. We've increased heart rate. So that's how we can regulate how fast the heart beats. Of course, when it's time to slow down, we have parasympathetic activity that comes down and blocks adenylate cyclase. Maybe somebody is on beta blockers for high blood pressure. Now we understand what's going on, right? The beta blocker is a drug that's going to come and block these beta adrenergic receptors. And by blocking that, I can no longer make adenylate cyclase, or I can no longer activate adenylate cyclase, so I can't make cyclic AMP. That is going to decrease sodium permeability through the funny channels and slow the heart rate down. Very, very cool system. All right, let's look now. Remember, these are all about pacemaker cells. Let's look at the other cell type that we're interested in, and that is the contractile cardiomyocyte. If we were to draw this action potential, what is it going to look like? How is it going to be different? Well, first and foremost, we actually do have a resting membrane potential. We start down here. But interestingly, resting membrane potential for this is about as negative as any resting membrane potential in the body. We're sitting down here close to minus 90 millivolts. This does require an external stimulus. We hit threshold, dries us up. We start to come down, but then we plateau. And then we come back down. Of course, there's no hyperpolarization because we're already about as negative as we can get based on the Nernst potential for potassium. And so that minus 90 is about it. How do we get to minus 90? Let's look at that part first. This, of course, is increased potassium permeability. How do we increase it? This is interesting. Of course, we have the normal leak channels for potassium, but we're going to add to that. We're also going to add a special, what's called an IRK. It actually can also be written KIR, which stands for inward rectifying potassium channel. I don't know that it's important to understand the detail of how this works, but very similarly to the funny channel, it's a leak channel, but it increases or decreases its permeability based on charge. And so these open up below threshold, and as they open up and increase that potassium permeability, below threshold, this is what allows the cell to really get negative for its resting membrane potential. So that's kind of cool. Okay, let's look at this next phase. Right before we hit, we'll throw threshold right here. Before we hit threshold, what's driving this? We said it was an external response, and that's true. But if we contrast it with what's going on in the neuron, the external response was a ligand opening up a ligand-gated channel, or something opening up a mechanical gated channel. Here, please recognize this is all about gap junctions. The reason this cell begins to depolarize is because its next door neighbor has had an action potential, and all of the ions going into its neighbor fill in and spread through the gap junctions into this cell. That's how the heart works in a coordinated functional syncytium. So these ions rush into a neighboring cell, leak through the gap junctions, and then trigger the action potential in this cell. Next up, the action potential itself. This one is back to normal, right? These are voltage-gated sodium channels. We know they're fast. We know they close when they get to the top, and that's exactly what we see. And what do we expect to happen? Well, up here we've got voltage-gated potassium channels opening, and that's why we start to get negative. But then we don't. Then we plateau. So what's this plateau all about? If potassium is leaving, shouldn't I be getting more negative? And the answer is yes. So what's counteracting the potassium? Well, we've also opened up L-type. L stands for longevity. So as opposed to the T-type that we talked about before that were transient, these L-type channels are a lot longer. These are voltage-gated calcium channels. And this is actually, of everything that we talk about today, probably one of the most important things to recognize. This right here, this calcium-driven plateau, drives what we call calcium-induced calcium release. And this is all about the strength, well, maybe force is a better word, the force of the contraction. This is something that makes these heart cells unique to skeletal muscle or neurons. With neurons, and certainly with skeletal muscle, we saw this with skeletal muscle, right? Think about the role that the sarcoplasmic reticulum played in containing all of the internal calcium stores. And then when the action potential went down the T-tubules, you lift dihydropyridine off of ryanodine, and the calcium would flood out into the cytoplasm and bind troponin. That would move tropomyosin out of the way, and then you get cross-bridge formation. You have your muscle contraction. The essential process is the same, but we see something different here. Whereas in skeletal muscle, dihydropyridine and ryanodine were coupled, cardiac muscle is different because we rely, right, because of what's going on here, we rely a lot more on extracellular calcium. We're not cramming all of the calcium into a sarcoplasmic reticulum. These cells have a sarcoplasmic reticulum, but it's not as well defined as you see in skeletal muscle because we're reliant a lot more on extracellular calcium. This actually adds an increased ability to regulate the strength of the muscle contraction based on where I'm getting my calcium from. And we'll show you how to do that in just a minute. So what do I mean by this calcium-induced calcium release? Well, if this was the surface of the cell, and we've got these L-type voltage-gated calcium channels, and as soon as this opens up, calcium is going to go in. Well, like we said, before, these were actually coupled. So this L-type calcium channel is dihydropyridine. It's in that same class. But they're uncoupled. We have to go deep within the cell to find down here my sarcoplasmic reticulum. Of course, this is loaded with calcium. And we have ryanodine as the channel that's going to let that calcium out. Calcium that comes into the cell can do one of two things. It can go bind to troponin, and of course this is going to give us cross bridge formation and contraction. Some of it will also come down and bind to ryanodine. That's going to open up ryanodine and let this calcium out. And of course, that calcium is going to contribute to everything that's going on with the contraction. So the key to understanding this idea of calcium-induced calcium release is more calcium equals stronger or more forceful contraction. And I can regulate the amount of calcium coming into the cell in part through how long this plateau is actually open. So we talked before about what epinephrine does to increase heart rate. Here, epinephrine's working in a slightly different way. Epinephrine's binding to beta-adrenergic receptors, and through a series of activation, it's all G-protein coupled receptor signaling again, but this is actually leading to modification of these L-type voltage-gated calcium channels, causing increased calcium permeability. And so epinephrine leads to a more forceful contraction in these cells because it lets more calcium in. Of course, these L-type voltage-gated calcium channels will eventually close, and when they do, what's left? Well, all of this, the repolarizing, this is going to be all done by potassium, right? The voltage-gated potassium that we were talking about, and then we're opening up these ERK channels. That's what allows us to get to this really low -90 resting membrane potential. The interesting thing, and the last thing that we want to add to this, because we're so reliant on calcium from extracellular stores, it is important for the cell to be able to get rid of that calcium. Before, we had the circa pump on the surface of the SR, right? We could even go back over here. And this is still here, right? We've got circa, and that's pumping in, this is ATP driven, that's pumping in the calcium back inside. But what about all the stuff that came from the outside? Well, we do have a couple of different ways of getting rid of that. The most common one is an antiport exchanger on the surface of the cell that takes calcium out in exchange for sodium coming in. So we use an antiport secondary active transporter to help get the calcium that came in from the outside back outside so that we can get back down to resting membrane potential. Of course, that sodium is going to be turned around and pumped right back out with the sodium-potassium pump. So hopefully that gives you a good sense of the different types of action potentials that we see and the different changes. The biggest changes in the pacemaker action potential, the contractile cell, or I'm sorry, the non-contractile cell, the funny channel driving the spontaneous depolarization and how it can be activated, regulated by cyclic nucleotides. And of course, the action potential itself is driven by voltage-gated calcium channels, T-type calcium channels. And then looking at the contractile cardiomyocyte, really negative resting membrane potential, in part because of ERK channels. Gap junctions are responsible for depolarizing the cell to reach threshold. Threshold is driven by sodium, voltage-gated sodium channels. The unique thing about this is the plateau. So at the time when we normally would be repolarizing, these L-type voltage-gated calcium channels open and counterbalance the efflux of potassium so that we maintain this plateau. And this allows a lot of calcium to come inside the cell. That calcium is super important because the more calcium I have, the stronger the contraction. Calcium-induced calcium release of more calcium from the SR, and that's going to really strengthen the contraction by increasing the number of cross bridges that are able to form.